Let's go to the last speaker, King Chow from the local institution. And the title is The Capture of Randomness, the Making of a Biological Slot Machine.
I'm going to talk about the capture of randomness. In fact, I think it's been mentioned in multiple times that about this particular project, probably because when we first submitted this project, we used a very fancy term. We call that it's a biological slot machine, right? So, but anyway, it is simply a report about the first steps. We are moving towards it, and we haven't got a slot machine made yet. So don't be disappointed at the end. Now, so I would ask, why am I here? We have heard Václav talk about his work, which is lasting for 60 years, covering lambda phage, T4 phage, all the mammalian cell, DNA moving around. In fact, I enjoy very much the talk because it reminds me a lot of things that when I went through the college years. I'll tell you why. And Drew talked about the work which was done in the past 6 years, and then moving forward, how all these biological parts should be fabricated and what the standard should be. Why am I here? So Václav, do you remember what it said? So it turns out that this is a paper Václav published almost 40 years ago with a person called K.K. Mark. It turned out that's the person that is my undergraduate supervisor. He gave me the key for his lab, and I keep a sleeping bag in his lab, and I sleep in his lab for 4 years. No, not 4 years, but sometimes when I have late nights, I stay there. And a lot of the experiment that Václav was talking about, KK taught me that too. So therefore, it reminds me actually what kind of work it is. Now, but moving on from that, during all the lambda phage, T4 phages, I move on and work on with worms. And since then, I get converted and always think that using all these genetics tools and molecular genetics tools, it allows me to understand a lot about biological function. Particularly, I'm focusing on pattern formation and organ assembly. So pattern formation, I'm looking at this tiny worm, C. elegans, looking at how the male animal, they are setting up their male tail with all the sensory organ. Each of them have a very specific property so that there is clearly a genetic program that controls how they're going to develop with specific features. And the second thing is that for each of these processes, they are made of only 4 cells. And I want to understand how these 4 cells get assembled together to become a functional unit. So the question that I have is really about a worm, how a worm is formed. And this is a picture taken from a very recent paper from the Star Institute, which is about a corn snake, and they're talking about somatogenesis. Why a snake is so long, and why a frog is so short. They have only 10 vertebrae. Now, the take-home message of that is, in fact, in this kind of animal, they're going through pattern formation, animal development. And there's 2 processes going on. One is long-range pattern, which is in this case referring to all this segmentation clock and how it's being coupled with the developmental clock. And the other thing is the local execution, whereas when the pattern is set up locally, there's something that controls how a somite is going to be formed. And of course, to me, as a developmental biologist to start with, I always think of pattern. When we have a zebra, why do we have all the zebra stripe? There must be long-range control of the patterning, how they get spaced out, and there must be local execution about how the pigmentation get deposited. Now, so with that, in fact, I work with worm, and then over the years, in fact, we work out a network of genes, how they get coupled together. So on the left-hand side, in fact, is a very well-documented BMP pathway, bone of genetic protein, which is controlling a whole cascade of transcription factors, which help to pattern the animal so that the animal know where is the front, where is the end. So that's a patterning question. But at the same time, when we look at each sensory organ, we also notice that there's a whole group of transcription factors which are interacting with each other in a network fashion with all this feedback loop and so on and so forth, and they control how an organ, these 4 cells, assemble together. So to me, I think the challenge is we always take genetics and molecular genetics as approach that we try to solve biological questions. But then it came to me that I find that maybe actually we should take a slightly different approach, and that's how I get started associated with the bioengineers and try to understand how would engineer look at developmental question. would they be able to look at it in a slightly different angle and give me some perspective how to develop it further? So what I see is that instead of doing all this experimental work, well, this is a figure taken from this particular review, we can go the other way. Can we simply start off with a very simple design of a circuit, and we try to organize it, and then we work out the model and see how it turns out and what will be the manifestation of that model. And we can contrast that with whatever we have as the experimental data and match it. If they match it well, then certainly it is something very useful. So what I'm going to talk about is not something for the past 60 years, not something which is just for the last 6 years, but it's only for the last 6 months. So I'm going to present the work, which we started off by trying to make a model and see whether we can obtain some experimental data so that we can fine-tune it further, and eventually we'll be able to perform a biological task, which will mimic something that we see every day. Now, so therefore, that's the synthetic biology, and so we started off with that, and in fact, is really initiated by the visit of Drew and Chris last year. And so we think about that. Well, maybe we should start with that and start with the iGERM and see at the infant stage in Hong Kong whether we can do something. And I understand that JD is here. So we have a Hong Kong U. They also have a team working on the iGERM. So I guess we're parallelly trying to do something and see whether we can make it. All right. So what I'm going to do is in the next maybe 10 or 15 minutes, I'm going to show you actually what we have done and what we try to do. Now, basically, what I see is that it's a whole exercise of practicing. So yesterday, we've heard students talking about how they went through the iGERM and what kind of great experience it is. I'm going to tell you that from an instructor's perspective, what we see is important. So I believe that is simply a run of practicing, and we try to gain experience with the participation of students, graduate students, undergraduate students, and have the high school students, and everybody's learning out of it. Now, so here is the team. The first thing that we talk about is integration, fabrication of parts. So we were able to establish a team integrating people together. So there are 3 high school students, Sunny, this is Vivian and Gladys, and they are all from different departments, including biochemistry, chemistry, physics, computer science, computer engineering, and high school science students. So we assembled them together roughly about 6 months ago. So we went through a lot of brainstorming process, and there were a lot of wild ideas that came out. But then the most important experience I had is that I was always overwhelmed by their enthusiasm, and it seems that they can conquer the world. And they have all the great idea that I can make this and I can make that almost instantaneously. And they would say that, well, in 2 months' time, I can do all this. And they're full of enthusiasm. Have they made it? Are they here? Now, so finally, after all this discussion, we came up with the idea, something that we wonder a bit. There's a lot of randomness going on. It's everywhere. We say that, well, so the random motion, when we talk about physics, so there's the thermodynamic properties of gas, so on and so forth. In computer science, communication theory, we talk about noise. Noise actually is randomness. And when we talk about biology, we talk about evolution. And evolution actually ascribes to the observed diversity of life, but initially it's because of the random genetic mutation. Okay? So randomness is everywhere. And even recently, when we have a market crisis, random rock hypothesis suggests to us for economy, there's no hypothesis. Everything is random. You can't predict. So what happened? Randomness. So the question that we have is, well, probably some of you would have it included in this trip. You probably will visit Macau, our big casino in the East. And when you get there, you'll see that there's a lot of slot machine. So you bet on your luck. You go there. Will you be lucky to get some money out of it? All right? Is it something you can predict, or is it a random process? Or I can predict, most likely, you're going to lose. So therefore, the students were inspired about this randomness. And they think about, well, if that's the case, if you go to a slot machine, you pull the handle, and then once you get it, you have a jackpot. So will we be able to generate some sort of complete randomness, a state of that, by design? Is it possible? So going through that, we say, well, is it possible? Definitely it's possible, but just how likely it is, right? So we try to boil it down to some question, and we ask, well, randomness, what it is. And in a biological system, how do we generate that randomness? So the randomness here we use is simply to monitor the random number in a binary format. And when it's in binary format, so it's all this 1 and 0, on and off state. So therefore, we ask whether we will be able to generate on and off, 1 or 0. If we can do that, then it would be great. And we know that, in fact, there are already existing bistable switch to tell us that you can turn it on or turn it off in the way that you like it. But then, using such a circuit, basically it depends on what kind of signal you provide to it. So it is going to give you a signal or give you no signal. So therefore, the inducer is very important. So that basically it's under your control. What's the randomness about it? So therefore, in our mind, we try to develop something like this. So basically, we want to generate a circuit like this, which, when the ball drops, it falls onto one side, and then it closes the circuit, and therefore, you see the light. Okay? And if this side is on, we say that it's 0. And if you do it again, this is an important part. This time, it may go to the left, and it closes the circuit, and then you get 1. Now, can we create a circuit like that? And so therefore, this is really the most important part. And how would this part be constructed? So the idea, taking that bistable switch forward, we say that, well, can we make 2 promoters going in opposite direction and see actually if I have, for example, a polymerase or transcription factor. When it's coming down, how is it associated with this promoter, whether it go one way or the other? Now, when we make 2 promoters like that, of course, we need to recognize that promoters, each of them, they will have their own efficiency. So if one side is stronger than the other, then you have a tendency that this polymerase always binds on one side. So therefore, it is no longer random. It's more predictable in that case. So therefore, in order to eliminate this non-randomness, we think about the following. Well, can we use the same promoter? And we simply put it there and allow it to work. So basically, the students thought about, well, use the T7 promoter, which is only 18 nucleotides in length. And if you have a polymerase that binds onto the T7 promoter, then it will turn on a reporter. Okay, that works. In this case, we also ask, well, can we create another T7 promoter, which is a mutant? Now, fortunately, in this particular position, when we make that mutation, this position corresponding there, they are overlapping. Now, if there is a mutation, what happens is that they would not work. How about if we flip them so that they are going in opposite direction? So if they're going in opposite direction, if they are well separated away from each other, then essentially you can have a polymerase coming in, binding on one side. You can have another polymerase come in, bind on the other side. Both sides will get turned on again. So that means you don't get any selection or randomness, all right? But on the other hand, fortunately, for this particular promoter at the last few nucleotides, when you connect them together, in fact, we can make them overlapping. So in this case, it becomes the key. That means when the polymerase come on and bind onto one promoter, it is not either going to be the left or going to the right. And it's a random process. And we hope to capture that randomness. So in fact, we're trying to do a whole bunch of variation of this overlapping and see how it's going to work. We're not there yet. So the idea is the following. If we can do that, then what now is we can throw the coin, whether you get the head or get the tail, is really depending on the polymerase. So you need to provide that polymerase. You need to throw the dice. So what we do is that we use an inducible promoter and try to turn on the T7 polymerase. All right? So once you can get that, so these are the construct that we're making and trying to address whether we can generate these components to allow us to assemble together. And once we have that, we have the T7 polymerase. That means you can use an inducer to induce the production of T7 polymerase, which will then bind onto this so-called randomized bidirectional promoter, and it's going to make a choice. And of course, together with this bistable switch, we want to have it, once it's transcribed here, we want it to come back and be able to inhibit the other side. So once it picks one direction, it will never go to the other direction. So we have that repressor binding site positioned there so that we generate that bistable effect. And we make these constructs and try to test out how they work effectively. Okay? Now, once you get that, that's our predicted outcome. The outcome would be the following. By the start, you add the inducer. The inducer comes in, results in the production of the T7 polymerase. This T7 polymerase is going to come here. So it may bind to the left, which will then transcribe, create a repressor, at the same time turn on a reporter. This repressor come in, bind onto this operator, so that this side will never be active. And all the time, it will be always going in one direction. Now, if you're using a reporter of this sort, this side is always using the GFP, and the other side is the RFP. What you have is then you will always get a cell which is green. So that's what we expect. Now, but then the problem follows. We know that when we make DNA, the T7 polymerase, we are using an inducer to induce its production. How many T7 polymerase is going to be produced? There will be many, right? It's not a single copy. So therefore, right now we're thinking about ways how to tune down the production of T7 polymerase to a level as low as possible, maybe at the range of a few copies, okay? We try to do that. And of course, there's a lot of hurdles, all right? And the second thing is the target number. Why the target number is important? Because if, in this case, instead of going in one direction, now you can have start again. It can bind onto the right. So what happens if, even though you have one copy of T7 polymerase produced, if you have two targets in the cell, one target may bind to the right, one target bind to the left. So within the cell, you would have both the green as well as the red. It's still a mixture. So therefore, we think about that, then it is not a good idea to do that. So when you have too many cones, or too many targets, what you get is only a mix of color. So therefore, randomness in that case is simply averaging everything, and it will not work. So the student thought about that, they went through the literature, and they identified a system which is using a single copy integration. So what they do is that they use this called lambda-inch system, which you can build a gene of interest on this particular plasmid, and then when it goes through the recombination, you would have this gene of interest replacing this part, the kanamycin-resistant component of the lambda phage, so that after the recombination, you would obtain this phage, and with the infection, after it goes in by the recombination again through the attachment site, this entire construct will be converted into something like this. All right? Now, bear in mind that in that construct, in fact, they do have a C1 TS mutant gene. So what happens is that once you have that in, if you switch the temperature to a high temperature, 42 degrees, then it's going to take out this phage part, and what's left behind is just this gene of interest. So now you will be able to obtain a single copy of this entire randomizer existing in the bacteria. Okay? So in that case, with one single copy, and any time when you have this polymerase coming in and bind onto the promoter, either it bind onto the left or bind onto the right. And once it turns on the green, then there will be a repressor to repress the other end. Then it will not be red. Okay? So that's the idea, and we're working on it. And in fact, thanks to the BioBrick Foundation that's providing all these bioparts, we are working on it, and we're working very hard on it, although a lot of you probably know, a lot of the parts, they don't work. And we were going through that process. So having that, we try to conceptualize the idea that having that circuit made as a randomizer. Now, we have a big ambition at the very initial point. We say that once we have a randomizer, we want to do something more. Randomizer only tells you 1 and 0. Random means a large number. Any number is possible. Can we make it larger? So what we have in mind is that we want to use the randomizer to generate a number. But then at the same time, we want the cells to be able to remember what the number is. So the idea is the following. So you can start and then enter the signal for the randomizer so that it's generating red at one point. How about if you start again? Okay. Okay, first you do it, you get one color. You do it again, you may get a second color. So when you have the first color and the second color being different. Now, of course, yesterday we did ask the students about that. How long does it take for you to push the button? Right? It takes hours. And then it takes a long time for them to really get color. So therefore, it takes time. But we can monitor these two consecutive events and say whether they are the same or they're not the same. Now, if they are the same or they are not the same, we want them to be read and be output as a different signal. So here, what we propose is that we want to have another component, which is called memorizer. We want to be able to remember what is the previous signal. If we can do that, then what we can do is to generate the inducer and then produce the first signal. And the first signal will be captured by this memorizer. And when you do it again, if it generates the same signal again, we'll say that, well, this memorizer would report it as I should not give you any reporter signal out of it. On the other hand, if the second round is a different color, then I will say, well, then because of the difference, then we will be reporting it. And that, we call it the jackpot. Now, we are thinking about this memorizer, and we are trying to adopt, which I'm not going to into detail because we are still working on how it's going to work. We're trying to adopt a bacterial tool hybrid system as a memorizing component to integrate them together so that we will be able to remember what's the last count as a color and what's the current count. So having that, essentially, that's what we want to do. If it all works, and if we can prove it in principle in 2 rounds of assay, that means we will generate 0, 0 to 1, 1. That means we can already generate from 0 to 3. And if we can prove it in principle, and if our memorizer, we can expand it further, that means it will be able to record even a longer strand of 1, 0, and 1. And in that case, we will be able really to generate a true randomness. So the question is, well, why do we want to generate a randomizer? Do we really want to generate a biological machine that gives you the randomness? Of course, no. Nobody's going to play that game because you throw the die, come back 2 days later and see what happened. But then, I believe that these are the things that how we see science work. I think 30, 40 years ago, when all these computer chips available, they're not working as fast as today. But then, if we can prove in concept that it works, in fact, we can potentially improve its efficiency and allows us to generate this randomness in a much wider application. And what we see is that random number is certainly very important. Currently, you look at a computer, they generate random number. How do they do that? They don't really generate a random number. They have a recorded number there, and they use a time scale to try to capture whatever information out of it. So it's not random at all, right? So here, we think that if we have such a biological system, then we would be able to capture the statistical sampling randomness. We would be able to simulate all this real complex phenomenon, processing the signal, even encryption key. And of course, people in Hong Kong would talk about if you can use this to capture the randomness of the market and how things work. So, are we there yet? No, we're still far away. We're still making those contracts. But what I hope is that in this presentation, I'll give you an idea actually what we're trying to do. And if you have other suggestions for us, it's much welcome. And I believe that we will be there. I'm not sure in a month we get the machine, but in the years, we will. So this is the whole team of people, and I'm simply standing here to present to you my motivation why I'm interested in synthetic biology. But then it's really this student who planned this whole thing out. All right, thanks.
Thank you very much. I was hoping you would tell me how to beat a slot machine. But I guess not. Anyway,
If I know, I would be in Macau now.
right. We have time for some questions. We're actually on time this morning. And the tea break starts in exactly
2 minutes.
3 minutes.
3 minutes, all right. Yes, Ruslan.
Instead of asking a question, I would like to thank you as a host here for this very nice symposium which you organized. And I forgot to mention at the end that you have helped me to prepare slides. And that, I guess, help you some idea, give how to use them.
Yeah.
So-
It's always great to see your previous speaker's slide first, and then you prepare your own slide.
So anyway, you ended with Lambda,
Yeah.
and that was the good point to end.
Yeah.
Excuse me.
Yes.
I would like to ask a question because you have 3 high school students. Do you think they can really understand what you're doing, or they're just working like for you?
First of all, they are not working for me. They are working for the team.
Yes, yes.
Now, the experience, in fact, I think it's a good time to share about that. For the high school student in Hong Kong, their training, actually, they're not really heavy on molecular biology. And they know about genes, but that's all they have. So therefore, when they come, in fact, our undergraduate students have a number of tutorials for them to beef them up a little bit to get them there. So I was really, I envy yesterday Alex's presentation about the Lincoln High School, that they have a biotechnology class in the high school so that they can do all the things there. But in Hong Kong, a lot of the high school, they don't have the opportunity. And it turns out that this 3 students, they've heard a lot about molecular biology, but they don't have the hand-on experience. They've never done it, and they don't even know the detail about it. So therefore, in a way, it's for us to teach them what it is. It's like giving them a crash course.
And since it's just for half a year they were together and making these stuffs, and but for a biology student in UST, for them to make such a thing, it takes them 1, 2, 3 years. So do you think there are any things to improve in our university education system so that in half a year all the undergraduates...
I hope that you asked that question yesterday when Roland was here.
Okay.
Yes, definitely. We need to do something. Right. Currently, I can say that a lot of the university courses offered in Hong Kong or even in the States, they would be a manual type of lab experience that the students are given a lab manual and they go into the lab, they follow the procedure, and they get everything done, and then they write a report out of it, right? But then I think in this experience, what I see is that the student really, they are self-motivated, they initiate the whole process of learning, dig up the literature, and so on and so forth. That's the kind of thing that we encourage. And in fact, we do have a small program in UST which is doing that, only for about 10 to 15 students, right? So that is something that we are promoting. So thanks for mentioning it. I hope that Roland was here.
One more.
Thank you very much for the very nice introduction of these two ways promoter running in two-way ideas. So it looked great. I just had one question because the gene on two sides of the promoter are very similar, and it might happen that homologous recombination will occur, and then the whole thing doesn't work.
That is a possibility. In fact, we haven't assembled the entire unit yet. We're still testing each component. We're making all these components. So therefore, that is a potential problem. We have that in mind. And in fact, when we pick the promoter, we have thought about that, and that's why we pick the short promoter, so that we even want them to be overlapping. But then other components, certainly there is a good chance that there will be homologous recombination.
And maybe one more question.
Right.
The second thing is that if you had such a very long gene, then probably you should integrate into the chromosome rather than use it as a plasmid, right?
At this point,
Yeah.
we're constructing it on a plasmid. But then at the end, we want to put the whole thing in a lambda page and then integrate into E. coli genome so that it will become a single copy. Right? So that's the ultimate. But at this point, we are still at the plasmid constructing.
Thank you.
Who invented the name iGEM? That's International Genetic Engineering Machines. Yeah?
Chris? Who invented it? Drew?
It's spontaneous generation.
Oh, randomly, maybe.
Random.
It just come up.